I'm not a great multitasker because when I'm talking in the class, I'm making eye contacts, my expressions. It's not easy for me to do it on Zoom. That's why I have the second Zoom account set up, and the camera is facing a different way so that you can at least see me in some way, but then I guess we all have to make adjustments. I don't know how I will try to be interactive in the class, and also Zoom, it's kind of tricky. I told in the first class, if you have something to contribute to the discussion, just unmute yourself and speak, because I can't keep track of the chat. So yeah, I don't know how I'll try, but then this is the best I can do, I feel like. So yeah, if you have an answer when I ask a question, just unmute yourself and start speaking up. I can't keep track of the chat. I can't see it on the window. I have to actively bring it up. Again, that disrupts the lecture. So the best way to contribute and participate in the discussion, if you don't understand something, unmute yourself and start talking. And then some of you have brought up concerns that I go off topic too much. Yes, I know that I do that, and sometimes it may be making things complicated in the sense that maybe it's often high level, but you don't have to worry about those things appearing in the exam. I'm not going to give you anything high level in the exam. Most of the time, what I'm trying to do is trying to give you a little bit of context, and also trying to give you the practical applications of those in society or in the clinical settings, just to get you excited about the topic. If I'm only talking about the concept without any practical application of those, what's the point of even learning any concepts? Like for me, it's supremely boring, I think. If I'm talking about, say, for example, acid froth staining, we're trying to distinguish mycobacterium from other bacteria. What are the implications of that? Like, somebody complained, why am I talking about the clinical things and why it's applicable? I mean, that's the whole point, isn't it? Why we study microbiology is because it has an implication on our personal life and in the society. I try to give you that just to keep you enthusiastic. I hope that's the message you're getting, and not trying to be too much concerned about those side of things and how that might appear in the exam. Like in the exam, I will be asking questions, like I'll be giving you scenarios. For example, those sample questions I posted, for example, like those images where the multiple choice style question, you should be able to use the concept you learned in the exam and be able to apply it to a situation. You're in the third year, I think you should be able to do that. For that, you have to obviously understand the concept. And if you don't understand the concept, just ask questions, or just come to the lecture, the office hours after, and they'll explain if you don't understand something. But there will be questions in the exam about the practical aspects of these concepts. I won't ask you a direct question what the concept is or technical details of it, but then you should be able to apply those to real life scenarios. Does that make sense? And I will give you context, and I will give you the practical applications of the concept I'm talking about in the class. And that's just to keep you excited about the topic, and not because I want you to understand that high level, like for example, I was talking about fluorescence microscopy, all the different stains. Yes, but I don't expect you to know, each and every stain and how it's used. Like I don't expect you to memorize any of that. Does that make sense? And yeah, same thing about like exams, like there won't be direct questions. There'll be some direct questions maybe, but then nothing of the sort that, directly about the concepts or memorizing any specific detail. There'll be more scenario type questions where you should be able to apply the concepts to a scenario that I'm talking about, and then you should be able to answer that. Yeah, I think, yeah, nothing too much. Like I'm not going to ask you anything that's totally different. That's nothing that's not being discussed in the class, or that's not on the slide, or that's not in the textbook. There's not going to be anything totally new. It will be on the concept that we've discussed in the class. It may be something that's not direct, where you still have to extrapolate your knowledge about the concept into a different situation, for example. Does that make sense? Any questions about those?
Hi.
Hi.
You said we do not have to know a lot of the depth of the slides, just the general. So are we expecting like some sort of questions that will be pretty straightforward to the slides?
Some questions could be straightforward, yes, yes. But there will be a lot of questions that are not so straightforward because I want you to be able to think, not just memorize things.
Yes, thank you.
Yeah, no problem. Anything else? Nope. Okay, so today we're going to move on to eukaryotic cell structure and functions. So we talked about prokaryotic cell structures, the different components and different functions of those. And then today we're going to talk about eukaryotes because a lot of the microbes are eukaryotes. So it's easy to think that eukaryotes are higher level organisms that you can see with your eyes, like higher plants and animals and humans, but then a lot of the eukaryotes, the huge components of eukaryotes are actually microbes. So for example, there are protozoans that are, a lot of them do cause disease. Some of them are just harmless. They're present in the environment. And there are called algae, there are single cell diatoms. Well, and a lot of them are just small microscopic animals like the tardigrades we talked about. So we are going to go through the different components of eukaryotic cells, their functions, and mostly focusing on what are the differences. Now that we've talked about the prokaryotic cell structure and functions, we should be able to kind of get an understanding of what's different with the prokaryotes. And then as we go through the different organelles and how they function, you will see that there's a lot of overlap in the functions between prokaryotes and the organelles of the eukaryotes. Again, supporting the endosymbiotic theory, a lot of the functions of the organelles and their structures, there is a lot of overlap between the prokaryotes and the eukaryotic organelles. So that's what we'll be studying this week. And again, we're not going to be going through super in depth, not like molecular mechanisms of how each of those things work, but then again, quite high level understanding the function and their structures. So like I mentioned, when we talk about eukaryotic microbes, there are protozoans, algae, fungi, plants, and animals. So we'll be talking in general. We won't be going through the cell structure of each of those different categories, but in general, the structure of different organelles and cell membrane and the nucleus and all of that. So the main characteristic that distinguishes eukaryotes from prokaryotes is the nucleus. So the prokaryotes don't have a distinct nucleus that's covered by a bilayer membrane, but in case of eukaryotes, there's the distinct nucleus with a double bilayer. So it's two bilayer membranes covering the nucleus. We'll go through in details about that, but that's the main distinguishing feature. The reason why they're called eukaryotes is they have a true nucleus. And then some of them are independent, like the protozoans and algae. So some of the single-celled organisms are pretty independent. The cell itself is the organism. So protozoans are great examples for that. And then there's also algae and some diatoms that are animals that are fully functioning organisms by themselves. And then there are multicellular organisms like plants and animals and higher order animals that are where the eukaryotic cells are part of tissues and organs and they're performing specialized functions. And then, so yeah, it has organelles obviously, and they're held by cytoskeleton. So we'll go through the details of how the cytoskeleton it acts as a matrix to keep all the cell organelles in place and also helps with the movement of the vesicles and movement of the molecules inside the cytoplasm. So cytoskeleton is another key feature that the prokaryotes don't have. And then the chromosomes, obviously. So again, we'll talk about that when we're talking about the nucleus. The prokaryotes don't have chromosomes, or you can say that whatever the DNA material that is present in the prokaryotes is one single chromosome. Often we call it bacterial chromosome. But in case of eukaryotes, there are distinct compact chromosomes, more than one. So humans have 46 chromosomes, 23 pairs. And in case of prokaryotes, it's always referred to as haploid or single. They're not paired. But in case of eukaryotes, they are paired, unless it's a reproductive cell, in which case the cell temporarily becomes haploid. Does that make sense? So they're all paired and they're diploid, but in case of eukaryotes, no, in case of eukaryotes, they're paired and they're diploid, and there's distinct chromosomes. They're rod-shaped chromosomes, but in case of prokaryotes, there is just the nucleus, nucleolus, which has no membrane, just basically circular chromosome. We'll talk about that a little bit more later on. And then when you talk about cell shapes, so he talked about in prokaryotes, they're rod-shaped or cocci, or there's spiralum. There's like there's a limited number of cell shapes, let's say. But in case of eukaryotes, you name it, there's a shape of that type of cell. Every possible shape. There's circular, long shapes, star-shaped cells. Like talk about humans, we have even the RBCs, for example, they're concave, they're biconcave shaped. Then they're also anucleated. So eukaryotes generally have one nucleus, but there are a few exceptions. Some of them, for example, the protozoan euglena has two different nuclei. But in case of prokaryotes, there's just a one nucleolus, and then there is sometimes plasmids, there's some loose DNA hanging around. But in case of eukaryotes, there is usually one nucleus, but sometimes there's more than one nucleus or nuclei. And again, diverse cell shapes. There's star-shaped, long, elongated, whatever shape you can possibly think of, there's a cell of that shape. So diverse cell shapes. So let's talk about the nucleus. So before I move on to nucleus, any questions about the introduction to eukaryotes? Okay, so nucleus houses all the cells' DNA, all as in the cells that play a role in heredity, and the nucleus is the DNA that's present in the nucleus is the one that controls all the functions of the cell. So this other DNA, for example, the mitochondria DNA, chloroplast DNA, those are the ones that help in the reproduction or helps in the division of the organelle itself, but not the entire cell. But the DNA that's in the nucleus helps in the division of the cell and the passing of the hereditary information to the daughter cells. And then it's covered by, so the nucleus has two distinct areas inside of it. There's the nucleolus, which we don't want to get confused with the nucleolus of the prokaryotes. So in case of eukaryotes, the nucleolus is the part inside the nucleus where there is production of the ribosomes. And so it's not the complete production of the ribosomes, the initial stages of ribosome production. And then the partly produced ribosomes actually flow out of the nucleus, and there's assembly of the ribosomes happens in the cytoplasm. And the nucleus is covered by a nuclear envelope. It is a double bilayer. So there's two bilayers surrounding the nucleus, and it is not continuous. It's got distinct pores that are guarded by these rosette-shaped proteins that help control the movement of molecules in and out of the nucleus. So you might have seen the structure of how the eukaryotic nucleus is drawn is always discontinuous. There's lots of gaps because there's lots of movement of DNA and mRNA and ribosomal RNAs, lots of in and outward flow of molecules from the nucleus to the cytoplasm. There's lots of pores. And the nuclear envelope, there is no separation between the nuclear envelope and the rough endoplasmic reticulum and the smooth endoplasmic reticulum. So endoplasmic reticulum is closely associated with the nucleus because that membrane pretty much forms the continuous system. And then the membrane system pretty much becomes the endoplasmic reticulum. Does that make sense? Yeah, any questions so far? Fairly easy to understand. And then the nucleoplasm contains the DNA, and the DNA is often referred to as chromatin because it is a distinct rod-shaped chromosomes like I'm talking about, but those rod-shaped chromosomes, you won't see them until the cell is undergoing division. That's when you will see the distinct chromosomes, but otherwise present as loosely packed, not loosely packed, but then it's still tightly packed, but then you don't see the chromosomes until the cell is dividing. You can actually see under the microscope how the different chromosomes are separating, and you won't see those shapes until the cell is dividing. But otherwise, they're still tightly packed, but then it's just like a compact structure that's tightly coiled by histone proteins. So it's histone proteins, and then the DNA is basically just super coiled all around them and then just present as chromosomes, but then you won't be able to see them, but then it's just present as a compact, packed, just DNA inside the nucleoplasm, the gel-like nucleoplasm. And then, yeah, I talked about the nucleolus, which is the area where there's rRNA biosynthesis. And the chromatin is basically present on the outside of the nucleolus. Oh, in case of prokaryotes.
Yeah. Like, is it a nucleoid?
Nucleoid.
Yeah.
Nucleoid, yeah.
Could they have a nucleolus with just a subregion of that nucleoid?
No, you won't see that. No, they don't have a nucleolus.
Okay.
Yeah. So all of the RNA biosynthesis happens in the cytoplasm in prokaryotes.
Okay. So it's not just the region of that nucleus or DNA where the RNA goes.
Within the nucleus. Yeah, yeah, exactly. Yeah. Any other question? Okay, so let's talk about eukaryotic ribosomes. So in case of prokaryotes, the ribosomes are 70S ribosomes, but in case of eukaryotes, they are 80S ribosomes, although the ribosomes present in mitochondria and chloroplasts are 70S, similar to those of the prokaryotes. But then the main cellular ribosomes that are involved in protein biosynthesis, they're 80S. And so the different subunits are 60S and 40S. So I had mentioned when we were talking about prokaryotic ribosomes is the S stands for Swedberg unit, and then it represents the sedimentation coefficient. So analytical centrifugation or ultracentrifugation, sucrose, when you do the sucrose gradient centrifugation, depending on the density of the ribosome, which depends on the type of proteins and type of RNA the ribosomes have, they sediment at different levels in the tube when you're doing sucrose gradient centrifugation. So depending on the density, they're giving all these different numbers. So the eukaryotic ribosomes are a lot more denser compared to the prokaryotic ribosomes, which is why it has the 80S ribosomes. And then there are two different kinds of ribosomes in eukaryotes. So there are the free-floating ribosomes, which mainly produce water-soluble proteins. And then there are ribosomes that are studded onto the endoplasmic reticulum, which form the rough endoplasmic reticulum. And that's why they're called rough, because they have all these ribosomes studded on them that are actively involved in protein synthesis. But the proteins that are produced by the ribosome that are attached to the endoplasmic reticulum, those proteins are usually destined for plasma membrane, the transport proteins, the glycoproteins that are part of the plasma membrane of the cell. So that's what they're destined for, or they're also involved in secretory pathways. That is the proteins that are secreted by the cell because it plays a functional role, for example, in case of immune cells. So we have cells that produce histamines, for example, because it has a role in allergy reaction. So there are cells in our body produce different kinds of proteins, right? So those type of proteins are produced by the ribosomes that are attached to the endoplasmic reticulum, which makes the endoplasmic reticulum rough endoplasmic reticulum. And there are cells that are mostly cytoplasmic proteins that are performing different enzymatic reactions, for example, or that are playing a role in metabolism. Those proteins are produced by the ribosomes that are free floating, they're free ribosomes. Does that make sense? And then, so because prokaryotic ribosomes are 70S and eukaryotic ribosomes are 80S, often antibiotics are designed to target each of those, so that if you have an infection, you want to kill just the bacteria because they're prokaryotic, they have a 70S ribosome. An example for an antibiotic that targets 70S ribosomes is chloramphenicol. So chloramphenicol is an antibiotic that targets just the bacterial 70S. But then it's a problem because eukaryotes also have 70S ribosomes, right? They're present in mitochondria, and in case of, like also in chloroplasts, there is 70S ribosomes. I mean, humans don't have chloroplasts, but then plants have chloroplasts, and they also have 70S ribosomes. So in humans, we have mitochondria that has 70S ribosomes. That's why often antibiotics have other side effects because of that. But then usually it's not a problem because the number of the 70S ribosomes is much smaller compared to how many there are of the ADS. So we still use chloramphenicol as an antibiotic. Any questions about ribosomes? Okay, so because we kind of already talked about endoplasmic reticulum, so let's talk about the ER. So the endoplasmic reticulum is an interconnected array of tubules. The tubules are called cisternae, which are just a flattened sacs, or the different layers. So the endoplasmic reticulum has a single bilayer, not like the nucleus that has a double bilayer. Endoplasmic reticulum has a single bilayer, and then, as I mentioned, they're studded with the ribosomes on the cytoplasmic side. The ribosomes are so that whatever the proteins that are produced by the ribosomes, so we know how the protein synthesis happens, right? So there's the two subunits and then the mRNA, and then the amino acids are added one after the other. There's a peptide bond formation. And then the peptide produced by the ribosomes that are attached to the endoplasmic reticulum is released inside the lumen of the endoplasmic reticulum. And then these proteins get released from the other side of the, so cytoplasmic side, and the one that faces the inside of the endoplasmic reticulum. So basically the proteins they get released from the endoplasmic reticulum as vesicles that are covered by the single bilayer of the endoplasmic reticulum. And then those vesicles then basically either directly get lodged in the cytoplasmic reticulum, depending on what level these proteins are processed. So once the peptides are produced by the ribosomes, there is a second step called the post-translational modification. Is that term familiar? A little bit. So these peptides are usually, you add like a secondary component to it, like maybe a sugar molecule, which makes the peptide a glycopeptide or a glycoproteins, because most of the transport proteins are present in the membrane that are glycoproteins, because they also have a sugar molecule along with the peptide. And then there's lipoproteins that have a lipid component added to it. So for the protein to be fully functional, once the peptide is produced, they have to go through a process called post-translation modification. And that step happens at the Golgi complex. So the endoplasmic reticulum basically produces the unprocessed peptide. So the ribosomes that's sitting on the cytoplasmic side of the endoplasmic reticulum produces the peptide, which enters the lumen of the endoplasmic reticulum, and then it gets released as a vesicle, because it basically covered by the same membrane that the endoplasmic reticulum has. And then the protein, the unprocessed protein, basically gets inserted inside these vesicles. And those vesicles basically then get transported to the Golgi complex, where they get post-translational, they undergo post-translational modification. Does that make sense? And then there's the smooth endoplasmic reticulum, which is basically involved in other functions like biosynthesis of lipids and carbohydrate metabolism and detoxification of toxic compounds within cells. So there's some sort of an overlap in the function between smooth endoplasmic reticulum and then lysosomes and peroxisomes. So we'll talk about that in a little bit. But so when you're talking about ribosomes, there's endoplasmic reticulum, there's the rough endoplasmic reticulum and smooth endoplasmic reticulum. So the rough one is mostly involved in protein synthesis for peptide, polypeptide biosynthesis. And the smooth endoplasmic reticulum is for the biosynthesis of lipids, carbohydrate metabolism, and detoxification of toxic compounds. And the cell is exposed to any sort of foreign materials. We're exposing ourselves to foreign materials all the time. And then along with the smooth endoplasmic reticulum, there is the lysozymes and peroxisomes that are involved in basically clearing up any of those foreign materials. Questions? Okay, so that will lead us to the Golgi apparatus. I kind of already talked about that a little bit, about the function of the Golgi apparatus. But again, it's an organelle that is basically made of just folds of membranous disc, and they're called dictyosomes, the different folds of the Golgi apparatus. And then again, it has a single lipid bilayer as opposed to the nucleus, which has a double bilayer. And then basically the folds are stacked together, and then it has a cis phase and a trans phase, depending on, so as I mentioned, the rough endoplasmic reticulum produces these vesicles with unprocessed peptides. They move through the cytoplasm to the Golgi apparatus, and then they get fused at the cis phase, because that's the receiving end of the Golgi apparatus. So the cis end, the vesicles basically get fused because it's the same lipid bilayer that endoplasmic reticulum has, and the Golgi apparatus is the same membrane. So the membrane, the vesicles, the membranous vesicles come and basically fuse at the cis phase of the Golgi apparatus. And then those peptides get processed to add maybe a sugar unit to make glycoprotein, or maybe they have maybe a lipid component, or they basically get folded through the process of post-translational modification. And now these active proteins get released from the trans side of the Golgi apparatus. And again, it gets released in the same way as a protein would leave the rough endoplasmic reticulum inside a single bilayer. So these vesicles then again get transported to the outside through the process of exocytosis, or they simply get fused in the membrane to make it the component of the membrane. It could be a transport protein, or usually transport proteins, or like a structural glycoprotein in the membrane. That's the main function of Golgi apparatus. Is that clear? So yeah, I mean, what makes the proteins undergo post-translational modifications is a series of different enzymes that add the glucose, or that helps with the folding of the peptides to make them into a functional protein. There are a lot of different enzymes, and then there are also obviously sugar molecules that need to be added to make the peptide into a glycopeptide or lipid, all the different components it needs to fold the proteins, or to add the additional component to make it into a functional protein. And then, yeah, and then as I mentioned, the transport vesicles, basically, they kind of bud off from the Golgi apparatus, and they get transported to the outside or to the membrane. Okay, so now we're moving on to the cytoskeleton. Any questions so far, or am I moving through pretty quickly? Pretty obvious, right? Yeah, functions are pretty straightforward. So we didn't really talk about the cytoskeleton when we were talking about prokaryotes, because they don't have a cytoskeleton. Mostly anything that's close to the biochemical properties of tubulins, for example, is the flagellum in case of bacteria. That's somewhat similar to the proteins and how they function. But eukaryotes also have a flagella, and that is made of tubulin. We'll talk about that a little bit, but the unique features of eukaryotes is presence of the cytoskeleton. And then if you focus in on the type of cytoskeleton, there's 3 different kinds. So microfilaments, and there's intermediate filaments, and microtubules. And they help with a lot of different functions. For example, in case of eukaryotes, we don't have a cell wall. So eukaryotes, I mean, except the plant cells, which have a cell wall. but animal cells don't have a cell wall. And what keeps the shape of the cell and gives it structure is the cytoskeleton. And each of those different types of cytoskeleton have their own functions, and the reason why they're called what they're called, for example. So in that figure, it's like I showed that picture in the last class too, but it's nice because it shows all three different kinds. So the microfilaments are also called actin. So it's basically two threads of actin just kind of wound against each other. But in this figure here, you can see the actin and you can see tubulin. I don't think you can see the intermediate filaments, but then I have another figure that shows the, or maybe there's a figure in the textbook that basically shows all three different type of cytoskeleton and how they're kind of functioning in their own way. So let's talk about the microfilaments. So as the name itself says, they're microfilaments. These are the thinnest of all the three different kinds. And then, as I mentioned, it's got two strands of actin or actin monomers that are kind of intertwined around each other to make into a fiber. And it's highly dynamic, meaning that it constantly polymerizes itself or assembles and gets disassembled constantly as the cell requires. And you'll understand why when you think about the muscle contraction, for example. So actin, in case of humans or animals, the muscle contraction happens because of the assembly of the microfilaments, and then there's constant assembly and disassembly. So if the muscle need to contract, they get assembled, and the muscle need to relax, it disassembles, and it does that with the help of myosin, another protein. So actin with the myosin helps in the muscle contraction. And if you consider the examples of the protozoan amoeba, so amoeba doesn't really have a shape in itself. It constantly changes its shape, and then the way it moves is by the formation of these false foot. I think that's what's called pseudopodia, which is it extends itself, and the way it's able to extend itself is because of the actin, because as it assembles, it forms into a fiber, and then it helps amoeba to move forward. And then actin or microfilament also forms the matrix all around near the peripheral region of the amoeba, or right inside the cytoplasm, and that's what gives amoeba like a temporary shape. It just keeps, so amoeba keeps changing its shape, and that's because of the actin, because of how it's able to polymerize and depolymerize constantly. The organism is able to change its shape and also move forward. Those are the main functions of microfilaments. Yeah, and the other function is it also helps with the cytoplasmic streaming, meaning if the cell needs to temporarily change its shape, even in case of humans, we don't have a distinct shape. All the cells change their shape all the time. And if the cytoplasm has to move towards like a newly formed edge, for example, and actin is the one that helps it do that. And then during cell division, so the formation of the furrow during cell division is because of the actin. Yeah, it's pretty straightforward. Any questions about the actin or microfilaments? Fairly straightforward. Don't think I missed anything. So yeah, next we'll talk about intermediate filaments. And intermediate filaments, you know, as the name itself says, kind of the size or the diameter of the fibers is kind of in between microfilaments and microtubules, about 10 nanometer, and it's thinner than the microtubules, which are the most thickest, and then it's pretty thicker than active filaments. So intermediate filaments are not made of one type of monomer. It's got different type of monomers. So for example, in case of keratin, the protein that forms our hair and nails and skin, they have the keratin protein monomers. And then there are desmosomes, which are made up of desmin. There are different type of monomers. They're diverse, made of diverse monomers. But then they obviously function, they have diverse functions as well as a result. And so they anchor the cells together in case of animal tissues. And if you think about the muscle cells, and what keeps the muscle cells sticking together is formation of these desmosomes. They're made of desmin. So the tissues are held together mainly by the intermediate filaments. And like I said, so there's the keratin of hair and nail and skin that are made of intermediate filaments. And another important thing is the space inside the nucleus. It's called the nuclear lamina, and that's what keeps the shape of the nucleus, and that's mainly because of the intermediate filaments. So usually the nucleus is surrounded by a bunch of intermediate filament fibers, which keep the nucleus in shape. And also it keeps the nucleus in a certain place inside the cell. That's because of intermediate filaments. The third type is a microtubule. So microtubules are the one that that kind of form the main matrix inside the cells. The microtubules are the one that keep all the cell organelles in place, and these are the thickest, and about 23 nanometers, so more than the double the diameter of the intermediate filaments. And they act as a scaffold of the cytoskeleton inside the cell, basically helping the movement of the vesicles from inside and to the outside, that's what helps with the exocytosis, keeps all the organelles in place, and that's all because of the microtubules. And these are made up of tubulin proteins, tubulin monomers, and so they work with other motor proteins like dynein and kinesin to move the organelles inside. And then, as I said, the vesicles that are released from the endoplasmic reticulum that move to Golgi complex, or ones that move to the outside of the cell, those are all facilitated by the microtubules. And then microtubules are also the main components of eukaryotic flagella. So there are some cells that are like protozoans, for example, they have flagella. Most of the protozoans that are present in aquatic environments they swim, and because of the flagella, and these flagella are made of microtubules. And also cilia. So we know the intestinal, the human gut cells are aligned with the cilia, and they constantly moving, and those are usually made of microtubules. So microtubules, the structure of those, you can compare them to the structure of flagella. They have a basal body, and then the filament, which helps the cell to move forward, and that is for single cell, the protozoans. And then so there's alpha and beta tubulin, which is the monomers, and it's like a structural difference in the alpha and beta. So the alpha chain and the beta chain. We don't need to go into the details of the structure. It's just basically how the polypeptides are arranged. You just have to know that they're made of alpha and beta tubulin monomers. And then, the monomers form basically the rod, which is the fiber. And that they form a scaffold inside the cytoplasm, basically holding all the organelles together, helps them with the movement of vesicles in and out of the cell. Yeah. What means, sorry? Scaffold? Oh, scaffold means basically like a supporting structure. Yeah, like a supporting framework or a supporting matrix. Or girdlers is the other term. I couldn't come up with a different term for girders, basically as a supporting structure, supporting cytoskeleton. Any questions? Microtubule, yes, yeah, that's the thing I forgot. So microtubules are also dynamic. So they keep changing. They keep polymerizing and depolymerizing because if the vesicles have to move, for example, they have to constantly form new fibers as the cell needs. But the intermediate filaments are more stable. They don't move as much. But the microfilaments and the microtubules, they keep changing. Constantly recycling the monomers, pretty much, yeah. Yeah. Yeah, you don't need to go too much in depth on how the polymerization happens. There's a lot of enzymes. It's a complex process. I think there's also a video that I was watching. I think there's also a AFM video where the enzymes, how the different monomers are added, and then the chain, it happens pretty quickly on the order of microseconds, how these fibers are formed constantly inside the cell. It's a very dynamic process. Like the muscle contraction, as you can imagine, it has to happen really quickly. And yeah, it happens in a fairly short order. And there's enzyme machineries that are ready to polymerize and depolymerize pretty quickly. Outside of the cell?
Yeah, sorry, or maybe I'm thinking of it wrong. But it's interfacing in between the two muscles. So that is forming the two cells together.
No, that's the intermediate filaments are the ones that hold the two cells together. That's the desmosomes, and which are made of desmins. That's the one holds the tissues together. But actin is the one that helps with the muscle contraction, which happens. So the actin is present inside the cell. So actin, along with another protein called myosin. Myosin also forms another different type of fibers that's only in human muscle cells. They act together, which helps the muscle to contract. So when the muscle needs to contract, it just polymerizes quickly and then forms a fiber. And then if you relax the muscle, it just kind of dissipates. Yeah, disassembles.
Internal to the cell is a physical contraction of the cell that has that contraction of the muscle.
Yeah, I would think that even, yeah, pretty much. I think it happens. What constricts the cell is the assembly of the actin. When the cell's contracting, the whole entire tissue basically constricts. I would think, I don't know, I'm using my imagination here.
And those intermediate filaments are just holding cells. So
No, the intermediate filaments don't contribute to muscle contraction. It just holds the cells together. Yeah. Okay, anybody has any other questions? Five more minutes. Yeah, so I wanted to have a separate slide for this. This is another main function of microtubules is it helps with the cell division. So we may go through cell division a little bit more in detail in coming classes, but then centrioles are the cell organelles, which basically produce the microtubules. And these microtubules are the ones that play an important role in cell division. So when the chromosomes, you might have seen these microscopic cell division. I'm not sure, but then when the chromosomes undergo cell division, so there's formation of the new chromosomes, which are destined for the daughter cells. When the cell is dividing, the microtubules are the ones that hold to basically, there's one centriole on one pole and the other centriole on the other pole. And the microtubules basically just hold the chromosomes and then pull them apart to individual pole. That's like another main important function of microtubules is they form these microtubule fibers that hold the chromosomes together and then kind of pull them apart. And then there's a formation of the furrow right in the middle, and there's a constriction of the cell. So microtubules play a really important role in cell division. Centrioles are also called centrosomes. which are basically the organelles that produce microtubules. Okay, well, that's all I had for today. Hope I didn't rush too much, but do we have any questions? If not, we're gonna finish a few minutes early today, usually late, but we'll finish up a couple of minutes early. See you on Wednesday.